And over to you.
Thanks so much, Sachin, for the nice introduction. And thanks so much for having me at this very exciting workshop. I already learned a lot. And I have seen that in most speaker slides, we had already in the last slides kind of what they think would be the future perspectives for the field of structural biology. And I think this is where my talk begins. And I think I mainly reflect what people have so far shown or what they think could be the future. And I will just go maybe slightly a bit more in depth. And also a word of caution that some of or many of the perspectives that I'm thinking about are antibody based. So there will be a slight antibody bias. I hope that's okay for everyone. I would also like to preface this talk that many of the things that I'm showing here are inspired by a blog post from Mohammed Al-Quraishi, who is one of the leading scientists in structural biology and especially machine learning biology. And he has been thinking a lot about the future of the field. So what AlphaFold and Rosetta Fold mean for the field. So some of the points I'm just reflecting here as well. So, and one point that he had, which I find really nice, is that these advances that we have now seen kind of are doing for structural biology what DNA sequencing did for genomics, kind of saying that before this, we were not even dreaming about, how can we answer any kind of biological question from a structural point of view. But now almost any biological field can start asking questions always also from a structural perspective. And that is also underpinned now by the aim that AlphaFold, for example, has with EMBL and EBI together, that they want to look at the structure or infer the structure of 100 million protein sequences and put this into this database, making it possible to kind of Google your structure in the future. And that is, of course, a major advance that will have repercussions to many fields: cell biology, evolutionary biology, immunology, and so on and so forth. And I'm just trying to look at all these fields and see what the future might bring. And this is also, and what the future might bring is also kind of reflected in the limitations that remain, even though we have made this large advance now in structural biology. And just to recapitulate what the remaining limitations are, is that so far we will not have a grasp, or do not have a grasp on protein-protein interaction. But this is also, we have heard this in the talks already, this is kind of the next milestone that the authors are looking into, both from the AlphaFold point of view and the Rosetta Fold point of view. For example, predicting antibody-antigen binding. What we also still are a bit unsure of is how can we represent the structure of intrinsically disordered proteins. Certainly, maybe AlphaFold can be used to kind of identify such region, but making no predictions about the conformations that are happening in those regions. An important caveat is also that we do not have yet validated whether these new machine learning approaches are actually also predicting the effect of mutation. And especially here, we're thinking of effects like different conformations that lead to different function or to different protein-protein interaction, for example. And to most of these points, I will also come later in my talk a bit in more detail. And then another important caveat is also that we do not yet have a grasp on the time dynamics of folding. So as we are all aware of, there are many proteins exists in different confirmations, but so far AlphaFold and Rosetta Fold only give us one confirmation and not the few or many confirmations a protein can exist in. So that is still something that we need to work on. And then also the position of any non-protein components found in these experimental structures is still not validated. And I think here also for the antibody field, for example, we know that there can be even protein inserts into the antibody, into the variable region of the antibody that can then of course change the structure and then change binding. So all of these very exotic but also very common things like cofactors, metals, and ligands is still something for the future that the field needs to work on. And now I will go to a few different futures. So I have called them future 1 to future 9. And here I'm looking at protein-protein interaction, which is then I think the next big milestone that the field will hit in the future. And if we just look at the structures that are currently in the PDB, you see here is the total structures, about 180K. And the growth every year seems to be constant, which shows you that experimentally there hasn't been much progress being done and really amping up the orders of magnitude that we can screen experimentally structures. That's why this in silico approach to structural prediction is such a game changer now, because now this will go up basically exponentially once we have validated further and see that the predicted structures are actually in, or which structures are in good agreement with experiments. And then of course, there are hundreds of millions of protein sequences that we know. Just for antibodies, there are billions of sequences that we know from the sequence, but not from the structure. So this is also something for the future to work on. But if we look at the protein-protein interaction, the number of data points that we have here is about 15,000. And some of them might also be pseudo-redundant, so quite similar to one another. These data sets also reflect a lot what people like to work on. So it's biased from a scientific questions point of view also. And there you see that the number of data points is already one magnitude lower than what we have for the structural data sets. And then although the problem of protein-protein interaction is even more difficult than just protein structure, so this gives you an idea of how difficult it will be to solve this problem with machine learning. So this will be very exciting how this will be solved. Yes. So then I think we have seen there have been many papers in the past in a lot of journals that have looked at protein-protein interaction networks based on the sequence, so not on the structure, just based on sequence interaction maps, so based on large-scale screening, for example. And there you get these large networks. And now I think the next step would be to actually recapitulate this in the structural space and to really understand how these networks are built. And to also understand to what extent actually do we need structure to understand biology. Now that we have access to that, we can actually answer that question: to what extent does sequence or structure actually help in the understanding of biology? I think that's also a very important question. And then we can go into different tissues, how these protein-protein interaction networks work there, or compare structures across different functions, transporters, structural proteins, enzymes, antibodies, and so on and so forth. So the possibilities are endless, kind of. And then, of course, from my point of view, a very interesting problem is antibody-antigen binding, which is still an unresolved problem. And this is a very difficult problem because antibodies, as Philippe has also mentioned this morning, can be basically generated de novo. So there is not this evolutionary template that we have from other proteins, which are more stable, which are also encoded just in one gene or a few genes. Here, antibodies are created by the recombination of different genes in a pseudo-random fashion. But the interesting thing is that the binding region on the antibody is usually only up to 20 amino acids long, and yet we know that antibodies can almost bind to any natural antigen or even synthetic antigen. And how this is done is by higher order sequence dependencies within these very small sequence space. And how to resolve that in the future, I think, will be hugely exciting. And we have also seen this morning that the interaction map of antibodies and antigens is different, statistically also speaking, from non-immune protein-protein interaction. So making it difficult also to infer the molecular binding properties from one protein family maybe to another protein family. So that will be very interesting to deconvolve in the future. Then one limitation that we have so far is screening for functional changes in variants and understanding that structurally. So what has been pushing forward the field of genetic screening a lot in the past is it's a technique called deep mutational scanning, where you can go through a protein sequence or a genetic sequence of that protein and mutate every amino acid or combinations of them, and then look at how this change affects binding or expression. So here it's, for example, from a paper that has looked at RBD, so SARS-CoV-2 protein changes and how this changes then binding to ACE2. And of course, what we would like to have from these changes, so from these screenings, we already get binding or expression and no expression binding or non-binding, but why is that so on the structure level? And I think there we will see a lot of advances really understanding from a structural point of view what these mutations do to the protein sequence. And how important that is also for the antibody field has been recently shown by Mason et al., where they have shown that, for example, one amino acid change can already make the difference between binders in blue and non-binders in black. And binders and binders to the same epitope, so to the same region on the antigen, can be very different in sequence or very similar in sequence. And to understand these higher order sequence dependencies will be very important. What I think is also very important from this deep mutational scanning technology is that we get negative examples. So we know when we do a certain mutation that a given protein does not bind or does not express well anymore. From protein-protein interaction, we do not actually get negative data. So we do not get, until now at least, for two different proteins, do they bind or not, and why don't they bind or not? And I think probably the protein-protein interaction prediction field will have to merge somehow mutational scanning and structural analyses together to kind of also create negative data for protein-protein interaction on the structure level. And that will be interesting how people will solve this thing, because for machine learning, in order to learn rules, you most of the time need negative data, and how that will be generated will be difficult. Also on the variant level, we have a field that is now exploding now, which is generative machine learning, which means that you can input a training data set into a generative machine learning architecture, and there are several of them, in order to then generate new sequences that are different from the training data set, but retain a certain function. And that can also be further complicated, of course, with conditional generation. So you want only to have proteins with a certain biological activity, or you want to make sequences that are completely different from your training data set, so that are not in the distribution of your training data set functionally, for example, or you want to optimize multi-dimensionally with different properties the output space. And what is happening currently is that people usually use either computational oracles to score the output generated data, and that is quite difficult because we don't have a good, we mostly don't have good computational functions to say this protein has this function. So what people mostly resort to is experimental oracles so far, so really expressing the protein and then checking for activity. And that is very low throughput. So in the future, it will be important to maybe use these new structural approaches to design better computational oracles to better score generative machine learning from a structural point of view. So for example, here, this was a paper that was recently published in Nature Machine Intelligence, where they made new protein sequences that should have a certain enzyme function. And here you see that that they tested 55 generated sequences. But of course, they could have, so in silico, they generated many, many more, right? But this experimental bottleneck that we have so far, hopefully we can kind of supersede this with also computational oracles in the future. And what is also very exciting is that they looked at the generation quality as a function of data set size. And now that we have potentially in the future access to many more structural data points, we can actually look at a question that we often get is, how much sequences do you need for a certain machine learning approach to work well? And that you can only do once you have enough data. And now we can finally start to answer these kind of questions, these very basic questions, but these very important questions. How much structural information do we need in order to achieve good accuracy for a given machine learning model? And for example, here, this is just another example where generative machine learning has been used for the structural generation of antibodies, for example, and then has been applied to a binder task. And so we think also that synthetic data and experimental data should go hand in hand in the future. So we need, so because you can generate as many experimental structural or predicted structural data as you want, you will never have, you will never be able to explore the entire space that you want for in-depth benchmarking. And for that, we think synthetic structural data generation is very important. So simulating data sets where you know everything, so where you have a ground truth, and then to use these data sets for machine learning benchmarking. And this is what we did with this simulation framework for antibody antigen binding, which Philippe had presented this morning. So please go to his talk. I think it should be available soon, and then you can see what we did here. And then we use this simulated framework to then kind of benchmark also with respect to training data set size, a generative machine learning approach to antibody generation, for example. So these are the things that you can do. We also, of course, looked at different encodings for antibody antigen binding interaction prediction. So I think in the future, synthetic data and experimental data need to go hand in hand in a kind of a feedback loop of benchmarking and then further refinement of a given machine learning model for a given machine learning task. One thing that I think that I also mentioned is this is our protein molecular dynamics that we need to get a better hand on. And as you know, proteins can exist in different conformations. And for example, antibodies can exist without being bound. It has been found that at least for some antibodies, they can exist already in the conformation that they will take when they bind the antigen. And getting to this good conformation is, I think, a major challenge in the future. And as you probably all know, also molecular dynamic simulation takes a lot of time, usually supercomputers. And if we could shortcut that with a machine learning model, that would of course be fantastic. And there is a lot of progress in the field on that, which was recently reviewed here. So I encourage you to read that review. So then, as I already mentioned, also we can now start to answer the question: to what extent does structural information actually add to any kind of sequence-based prediction accuracy, right? Since, for example, we know for the antibody field that there is also information in the structure, and we know also, of course, that antibodies can bind to many different antigens, and each antigen can be bound by many different antibodies. And these higher order dependencies, how do we resolve them? And how does structural information help to resolving these dependencies? And we have recently kind of provided preliminary proof of principle. I would say that pairing sequence and structure together increases prediction accuracy of antibody-antigen binding. With larger scale data sets, that would even be, it would be even better to validate this prediction. And of course, this same kind of principle can also be used for any other protein-protein interaction approach, for example. So then something that we're really passionate about also is providing, so what comes after a predicted structure, right? So once you have these hundreds of millions of structures in the EBI database, you want to then use these structures to perform different machine learning tasks to kind of classify, why does this protein have this enzyme function but not this, for example. And for that, you need like a principled way of doing that. And we previously or recently put online a machine learning framework for immune receptors on the sequence level. But what we why we did this is because in any machine learning approach, in order to make it reproducible, you need a formalized way of defining your machine learning task, and you need a formalized way to perform the training, validation, and a test. And many things can go wrong there. So we think in the future, these kind of frameworks should also be available for the structural field and also for the structural antibody field, in order to also perform any kind of post-AlphaFold machine learning approach in a formalized setting. And yes, so for example, here there are a lot of different approaches already being applied to antibody machine learning, and we imagine that in the future also these kind of approaches could be performed within, for example, ecosystems such as our ImmuneML framework. And what we are also very passionate about is that these kind of frameworks can be applied to by non-experts. So we also have a graphical user interface where non-machine learning experts can perform machine learning analyses. Yes. So I think coming up with these frameworks will be hugely important. Then protein design will potentially be also be revolutionized. So here is an approach by the David Baker lab, where they use Rosetta to kind of take random sequences and then apply rules from 3D structure prediction, and then make these random sequences into sequences that are actually taking a native fold. So kind of answering the question of generating new sequences to fold to new structures. And once we have more data to train these models, we can also get better at really de novo protein design. So, and then I would maybe just go a bit faster now. So I think we have talked a lot about antibody drug discovery, so I don't have to go much into detail, just maybe one thing. So there are already deep learning approaches for antibody structural prediction. And the problem with antibodies is that they can have these very long loops. And the longer these loops are, the worse prediction accuracy is. So I think in the future, it will be very important to especially predict these long loops. As for example, it has been shown that broadly neutralizing antibodies, not only here by cows, but also by humans, are actually quite long. And how do we get to that will be very exciting. Then in antibodies, you also have not only binding specificity, but also developability, so different physicochemical properties that you want to optimize, and all of them lie also in the binding interface. So we need also in the future a structural way to really at large scale design antibodies in a multi-parameter fashion. I think one field that will also be hugely impacted is evolutionary biology, because now you can actually start to answer questions like how are proteins conserved over time, not only based on structure, but also on function. Here's a paper where they have looked at structures across billions of years, but that was only for a few structures. Now we can do this for many, many structures. And what are the principles that are preserved or changed during evolution across organisms, and so on and so forth. So the questions are endless. And also from an antibody point of view, we know that a few changes in the antibody building blocks can change antibody function, which are called haplotypes, these small changes in the sequence. And how do they actually pertain to the structure and then to function, especially with also the emergence of precursors to broadly neutralizing antibodies for HIV or flu or SARS-CoV-2, for example. I think that will be very important to understand, and how these variations differ across different ethnicities, and from a structural point of view, how is the sequence to structure relationship? That will be very important to understand. And then my last slide is, so I think we are all thinking about, from a machine learning point of view, we want to look at the rules. But what do we mean by rules? So probably we mean by rules these higher order sequence dependencies that command folding or binding. And how do we get to those rules? Because only with those rules, and only maybe once we understand them better, we can actually rationally design proteins at a larger scale. And we are actually looking into that by building a formalized framework from a linguistics point of view, to actually come up with a formalized protein linguistics framework that can complement current NLP approaches, such as, for example, the BERT approach that has recently been published, where and it has been shown that BERT models are really successful in interpreting and really understanding protein modeling. So, for example, so these machine learning models are based on using attention, and it has been shown that attention correlates with these higher order sequence dependencies. So amino acid pairs that are close in physical space but lie apart in the sequence. And it has been shown that they correlate well with contact maps and also binding sites. So how do we really use these contact maps then to visualize and to extract these rules from these machine learning models and then map that back to structural rules? I think will be one important task for the future. And with that, I would like to thank everyone, and I'm open for questions.
Thanks, Victor, for the wonderful presentation. And I must also share with everyone that you have also contributed in the background by giving us, testing capabilities initially. So your contributions to this workshop has been immense. So we are grateful to you. Just to be concerned with the time, I think we do have questions. Can I request you if you can type the answers in chat while we may proceed with the program?
Sure. Thanks so much, everyone.
Yeah, thank you, Victor. Thanks again. Now we move on to the next talk, which again, like the first talk of the day, is joined by an expert from US. And I believe it's 5:30 a.m. In your place, Jim. So a very warm welcome from our side, from the Norwegian community, but also a lot of people joining us from all over the world. So we are very grateful to you for making this contribution. And I believe you're also joining us later in the panel discussion. So we really look forward, hearing from you about your vision and viewpoints on what's happening in the field. But before we begin, I don't know if you want to just quickly check if your slides are coming up right or not, and then I will introduce you.
Let me, can you see the screen now?
Yes, we can see it. Maybe you just go full screen and then just you can run the slides.
Let's see. Which view are you seeing? I can't.
Yeah, we can see them well. I think you just need to probably move them, or yeah.
Okay, hold on. I'm trying to get it into the, oops. Are you seeing which view are you seeing? I can't, I'm in an unusual place.
The normal view, which is for the audience. I don't know what view you have out on your side, but we see perfectly fine. We see the first slide probably.
Okay, I'm good then. It's not quite in the same state as normal, but I'm in a different place with a different setup than normal. so as long as you can see it, that's important. Thank you very much for the invitation to speak today. We've been working on collaborations with Norway, and particularly the cancer cluster, for a number of years here at Lawrence Livermore Lab. So my name is Jim Brase. I'm the Deputy Associate Director for Computing at Lawrence Livermore National Lab. So I manage all of our R&D around large-scale computation and its applications. My particular interest is in the area of applying high-performance computing and large-scale AI to biological problems in general. For the past 5 years now, almost, I've been working with a group to lead the ATOM Consortium, which was started up as a partnership as part of the cancer moonshot work by Vice President Biden at the time a few years ago. So I'm going to describe that work, what we're doing, and point out at least a couple of the connections potentially to work that's going on there as well. So I'll just jump in. So ATOM is really focused on looking at new approaches to make drug discovery faster and more effective. Today the times to go from an identified target to get through to a molecule that's ready to go into a human for the first time averages about 6 years. We'd like to find ways to make that significantly faster. We are focused on that initial part of the process, going from target to first molecule. Our goal is really, can we accelerate that process to allow us to get through that discovery and optimization phase in weeks, not the years that it takes today. And we believe that by taking new approaches that are driven by computation and making use of computational models that incorporate both mechanistic information and new machine learning methods, we can actually do that and focus down to the experiments that are necessary to make that process run. That's really shown here. The idea is to use an active learning workflow where we propose molecules, we predict their properties using a variety of models, we analyze those predictions for their uncertainties, and we can go around that gray in silico loop of optimization, prediction, and then rehypothesizing new molecules very quickly. And then when the uncertainties in the predictions grow to a certain point, we actually can break out of that loop and go actually do the specific experiments that are necessary to optimize our models and to increase the certainties in the models, and then restart the computational optimization loop. And by going through this process systematically, we can actually get down to the point where we're only doing the critical experiments. Early on, we're exploring and building and making the model work better. And then as that process converges into a part of chemical space, we're actually then validating that through those critical experiments. So it's still very much an integrated and combined computational and experimental process, but we're trying to make sure we spend our time on the critical experiments that really provide exactly the information that the computational models need. So we have three top level goals in ATOM. One is to actually demonstrate that process, that it works, that it's effective, and we've made a lot of progress along that line. Second, we're doing this in an open source public system. So we're developing, validating, and then releasing all the software for doing this. We're working with university groups. We're working with a variety of companies, large and small, but we're producing basically an open source platform that other groups can then take and build upon for their purposes. And then finally, within ATOM, we are in our partnership, we're applying the platform to making molecules, focusing because as I'll show in a minute, one of the founding partners is the National Cancer Institute. So cancer is a big focus. Of course, over the past couple of years, there has been a growing focus on working on infectious disease, and particularly SARS-CoV-2 therapeutics. But in the long term, within the actual partnership itself, we'd really like to then be using and exercising and validating and growing the platform by working on some of the large set of underserved needs for the public good. So ATOM is a public-private consortium. The consortium itself we consider to be an experiment. It's looking at, can we have a number of government entities work together with the private sector to really expedite and speed this up. So the founding organizations in 2017 have been Frederick National Lab for Cancer Research. They're part of the National Cancer Institute, University of California at San Francisco, and the Helen Diller Cancer Center at UCSF, Lawrence Livermore National Lab, where I work, where we bring the computing and the data science and machine learning to this process, and then GSK, who came in and they're with their leader John Baldoni in this area to bring in the long experience in drug discovery and they provided early on a lot of data to sort of bootstrap ATOM up to where we are now. Since then, there have been quite a number of additional partners and collaborators who have come in. We have 3 additional national labs in the US, Argonne National Lab, Oak Ridge National Lab, and Brookhaven, who have officially joined the partnership and are providing people and capabilities to what we're doing. And then a number of other partners working, a number of other collaborators working at smaller scales with us to enhance the computing that we're doing through NVIDIA, for example, to increase the data sets that we have with Acceler is one of the largest data companies in pharma. University partnerships where we're providing actually the open tools and data sets and models to classes in pharmacology and from pharmacology to AI classes. So quite a number of groups are using some of the open tools that we're providing. We're working with Lawrence Berkeley National Lab on actually applying this design process that we've developed for molecules, but applying it now to biofuels, not to pharmaceuticals. So the process, by changing the data sets, changing the models that are used for the properties, can be applied to a number of molecular design problems. And so that's an interesting and exciting area. This is a little bit more detailed view of ADAM and how the workflow that we use. It's really based around a working compound library shown in the top left. These are the same two loops, the gray loop and the yellow loop, that I talked through at the higher level a couple slides ago. But typically in that working compound library, we'll have around 100,000 molecules that we're working on at the same time, so working on them all in parallel. For each of those molecules, we will compute a number of properties, usually multiple efficacy properties, how the molecule interacts with the particular target protein or the target phenotypic assay that we're using. A number of safety properties, how it interacts with other targets that we don't want it to. Pharmacokinetic properties like solubility, permeability through various membranes, clearance rates from the body, things like that. And then developability properties. How hard is it to actually synthesize this molecule? Does it have have some of the stability and so on properties that we'd like to see? And so we predict typically around 20 properties of each molecule, compare those to the design criteria, and then the generative molecular design block here actually then projects those molecules into a learned latent space of a generative model, like for example, junction tree autoencoder is the one we generally use. And we actually do then the optimization in that continuous latent space, project back out into physical space, and then go around the loop again. So that's the set of computations that we do. We have lots of information on that in extreme detail that we're happy to share with people. The yellow loop then is shown here, where the active learning component is analyzing the uncertainty in the predictions and making determinations when we need to do experiments to provide additional data and what those specific experiments should be. And then that data is used to update the predictive models. It's really managing the applicability domain of the model. So where in chemical space are the models able to make good predictions? The optimizer is changing them and moving them in chemical space. They eventually move away from the training sets. And so as we run, we need to bring in new data to update those models and to move the applicability domains into the place where the optimizer wants to go. So ADAM produces a number of specific products. We have an open molecular data library where we're pulling in data from a variety of sources all around the world. And we curate that and set that up so that it's actually ready to use in the machine learning models that we have. The second product is called AMPLE. That stands for the ADAM Modeling Pipeline. That is the system that actually trains the property prediction models. It optimizes them and then actually performs the property prediction models for the GMD process that I'll talk about in a minute. That's been released open source. It's being used in I think the last I saw about 5 or 6 different university programs around the country here for students to learn how to apply machine learning to drug design. We're using it all the time in this. It runs on everything from our very large high performance computing clusters here at Livermore, where we can do extensive hyperparameter optimization on the models. But there are also cloud implementations that run on AWS and so on. That's what we typically use for student classes. LBAN is the third product that we're producing. It stands for Livermore Big Artificial Neural Network. That's actually used, that's an again, a released open source code that is used to train the large molecular generative models, the autoencoder structures that actually hypothesize new molecules based on the design errors that it sees at any given time. So that's a very large model, typically can be trained on up to 2 or 3 billion molecules, and runs on our large scale clusters here. Then GMD is the overall software that runs that gray computational optimization loop. As I said, it operates on typically around 100,000 models in parallel and manages the process of running all those property predictions, and then manages the projection and reconstruction of molecule and optimization of molecules in the generative molecular design process. That is in the process of being released openly now for the first time, but isn't available yet, but will be in the next 6 months, I think. And then finally, we actually run pilot design projects, and I'll talk more about those in a couple minutes. Adam uses 4 distinct predictive modeling methods within those predictive models. As I think I mentioned, we use both mechanistic modeling and data-driven machine learning modeling. So we're using traditional structure-based docking that's used in the initialization and setup processes to initialize that molecular population with sort of our best guess at molecules that might be appropriate in the particular part of chemical space for a particular target. We use physics-based simulation, molecular dynamics, to then refine those models, rescore the top docking hits. We can use both those approaches actually in the loop as well. So they're used both for initialization and actually in the design loop. We're beginning to use more and more now structure-based models based on machine learning, where we're taking the target structures and target and ligand co-structures and using that to actually predict binding for both efficacy and safety targets. And that's a method that is growing in use, and we actually have that system now built into our design loop as well. And then, but probably the main thing, particularly for safety and PK models and so on, are ligand-only models based on experimental data. So we have many fairly large data sets that are then just directly trained to predict the output of either biochemical binding assays or various phenotypic cell health assays or various PK assays. We do work in cases where we have very limited data sets there. We are working on things like physics-based simulation to augment those data sets. That's an active area of research as well. So we use all of these modeling methods, but a lot of them are structure-based and would be just a natural place to, for example, to collaborate on what's going on with AlphaFold and new machine learning methods for structure prediction as well. And we'd be very excited about working together in areas like that. Just to give you an example of the kind of models we're doing, we typically have, I said we typically optimize on 20 models, but we can use much larger numbers than that we wanted. And in a full drug design run, we will use larger numbers of models. But we have typically safety panels, so sets of collections of safety targets of 50 of them or so, and there's some examples shown here. And then, a fairly large set of pharmacokinetic models, both in vitro models, in vivo models. The in vivo models tend to have small data sets. So we use sometimes different modeling methods for those, but this is just to give you a flavor of some of the types of models that we're using in the design process. We're also moving ahead with research on very advanced modeling methods. So this is an example of a method where we're actually analyzing the interaction between mutated RAS protein molecules, so important in areas like pancreatic cancer and such, on the membrane, and understanding that interaction and so on is opening up new potential targets for drugging RAS, but the actual modeling method that we're using here is one that we believe is applicable to a number of new areas. So the idea here has been that we're actually running MD, so molecular dynamics simulations at atomistic scales to understand the interaction between RAS and the cell membrane, but we're actually driving that then from a macro scale continuum model with a machine learning algorithm, then instantiating the atomistic molecular dynamics simulations when something interesting is happening. And that's actually allowed us to extend the time frame that we can do an effective molecular dynamics simulation from instead of a few nanoseconds to up to a millisecond. Now we don't, so it's basically allowing us to disregard all the sort of the low level thermal vibrations in the system and look at only the interesting parts of the interactions. And it's a whole new method of integrating machine learning with high resolution molecular dynamics to effectively extend the timescales that we can simulate and to get into biologically interesting time durations of the overall model. So we're very excited about the method and are working this year now and actually starting to apply that to some specific drug design applications as well. Finally, I wanted to show you a couple of the experiments that we've actually done. We've run a number of cases where we've done overall designs. This is one of our first cases that we did, designing a selective aurora kinase B form inhibitor. Aurora kinase is a kinase, a signaling protein that's important in cell division. It's an important cancer target as well. There are two basic forms of it, the A form and the B form. We typically want to inhibit the B form, but not inhibit the A form. So we've done here's a demonstration optimization that we did. In the upper left, we show the loop plus the actual criteria that we used. And you notice the B to A selectivity and the second efficacy metrics. We want the B to A selectivity to be 1,000 if possible. And then in the plot below, we show the A efficacy versus B efficacy. And each dot is a molecule. The red and blue dots along the diagonal where A inhibition and B inhibition are roughly equal are the initialization point, the initial population of molecules that we started with. The green dots in the upper left then are the top 200 or so of the final population of molecules. And you can see we actually moved in the direction as the optimization proceeded of greater B and then A inhibition, which is what we wanted in this. On the right, we show basically experimental validation data that was done. We took those 200 predicted compounds and actually made them and tested them and found generally results that matched pretty well between the model predictions of their properties across the safety and efficacy and PK parameters with the predicted values, and found quite a number of molecules in there that got at least very close. I'm not sure that any of them actually met every design criteria, but out of the 100,000 molecules we were doing, there were the population of molecules that were very close to meeting them was highly enriched. which is exactly what we're aiming for. So we've had good experimental validation of the method. Finally, we've been doing more of those kinds of design investigations. This is an example of an H1 antagonist design. This is basically building a better antihistamine. This is again an engineering example because there was actually no commercial interest in doing this, but we wanted to redesign antihistamines to be sensitive to inhibit H1, but not to inhibit muscarinic proteins, which cause drowsiness and side effects like that. And here we were actually looking at the timeline of how long it took us to do these things, and demonstrated that we could go, even still at an R&D stage this year, that we could go from the specification of where we wanted to work to getting data from our partner on this Neurocrine, to actually then running the optimization and synthesizing molecules in a robotic synthesis process in a few months, as opposed to a few years that this kind of thing would normally take. So we were very pleased with the progress in the time duration that this takes as well. Then finally, I wanted to show we're not just applying this to small molecules, we're also applying this to antibody design. Here's an example to SARS-CoV-2, where we've actually used the same computational processes of hypothesizing structures now for the sequence that corresponds to an antibody, then predicting its properties, its interactions with viral targets, and then particularly the spike protein in this case, and then optimizing that structure computationally, showing here that we used these are very large scale computations, because this is mostly a mechanistic prediction of the interaction, where we're using about 1 million GPU hours per iteration of the computational loop, then go out and actually make about 100 antibody mutants and experimentally test them versus the SARS-CoV-2 virus, and then repeat that operation. And we've actually been able to show that with typically about seven or eight mutations on that. The initialization here were SARS-CoV-1 neutralizing antibodies that did not bind SARS-CoV-2. So we started out with an antibody that we thought was in the right ballpark, but then actually mutated it at 7 sites here to actually give it SARS-CoV-2 neutralization. Binding first in the plots on the right. In iteration 1, these are plots actually of neutralization of the actual viral infection. In the first iteration, we typically get binding. In the second iterations, we're actually getting neutralization of viral infections using the pseudovirus in this case. These are not full up live SARS-CoV-2, but we have done it now on the full virus in our BSL-3 lab here at Livermore, and it works pretty closely to this. But so this is very exciting, and I think, as far as I know, was one of the first sort of full up computational designs of re-optimizations of an antibody to increase its binding and neutralization in SARS-CoV-2. We're actually now working with a number of antibody producers around the country to apply this optimization approach to therapeutic antibodies that for the initial variant and re-optimizing them for a number of the emerging variants that are coming out now. So this is being used quite heavily for that. Then finally, I just wanted to show that we're also on the bottom here. We're starting to work on a number of new cancer design projects, in particular, redesign of PARP inhibitors to enhance brain penetrance. So where we're taking a known drug, an effective drug, but re-optimizing one property. In this case, its ability to penetrate the blood-brain barrier. And using the same modeling and optimization approach to basically fix one problem with a particular molecule. And we are now actively working with a number of groups to develop new design projects. We're at the point now where the basic methods, the computational frameworks and so on work. And we're making progress now by actually applying this to molecular design projects. So there are opportunities to work together to do that kind of thing as well. I think that's my last slide, and I'll stop there. And if there is any time for questions, I'm happy to answer.
Yeah, indeed, there are some questions, and I'm sure Klas is also waiting to engage you, Jim, in the panel discussion. And excellent presentation to see state-of-the-art and cutting-edge work that's happening. And it's also good to learn that you're collaborating with Norway, and I believe it's Oslo Cancer Cluster is one of them.
Yeah, that's right.
So that's very good. So there is one question that has come from someone, maybe it's more of a generic question, but I think it's important. They're asking about the advice for mid-career scientists in the industry, because we are showcasing so many methodologies and frameworks that how do they keep up? How can they train themselves?
Right. That's a really good but very hard question too, because you're right. We're bringing together so many new technologies that the partnership aspect of this, I think, is really important. But I basically give the advice that I generally give, that I think getting comfortable and understanding at least some basic level of the computational modeling approaches in this, I think, is really important. Not that you need to be able to work to run, highly optimized models on supercomputers and things like that. We have there are people who are real specialists in that kind of area. But I think being comfortable with the application of computational modeling across a lot of these areas, I think is very important, because the growth in the application of those methods is just everywhere. It's not just in doing drug design, but in anything where we're working with data sets coming out of experiments and so on. The ability to apply some of these modeling methods and so on, and it's not really that steep of a learning curve, I think, to go from basic sort of statistical modeling that I think most scientists are comfortable with now, but to applying some basic machine learning methods to that, I think increases the power of that a lot. And it's not that big of a leap beyond that. So I'd recommend getting comfortable in that area, at least with the language and so on, because it's going to be a dominant theme, I think, in the future.
So is your lab also involved in organizing any specialized trainings or any such interventions for now? Or?
Not Livermore so much, but Frederick National Lab actually has been the partner within ADAM, which has really been organizing a lot of the outreach to universities, setting up of classes, because National Cancer Institute has a very strong training mission as well, and outreach and so on. And so I would say that would be a great thing to talk about. I would bring in my partner, Eric Stahlberg, who is my co-lead for ADAM from National Cancer Institute, and he would be happy to go into that much more deeply. But yeah, but we are actually organizing and running training and classes on, for example, like the AMPLE training pipeline that I mentioned earlier.
Thanks, Jim. We are having a lot of questions coming up now. Maybe with your permission, I can take two more. Mm-hmm. And we are eating up in the panel time right now, but I think they're a bit technical here. So first question is from Pedro Matias, which says, which SARS-CoV-2 variant have you tried? Do you think it is possible to develop a universal AB?
I can't answer the first question specifically on which variants have been worked on. I can connect you to the people doing it if you want detailed information. But they are actually attempting to do exactly what you suggested, which is to look at whether using these optimization methods, we can actually re-optimize antibodies so that to be as broad spectrum as possible with the kinds of mutations that we see. So there are models of what likely mutation sites are and so on, and we know we have some idea about what emerging variants will continue to look like. And so we are actually working on optimizations that we believe can actually be fairly broad across variants. I think we're pretty confident that in variants on a single virus like this, that we can be effective in finding optimizations for that. Whether you can then extend that to having one antibody, or the flip side, one vaccine that is like a pan coronavirus therapeutic, that I think is much harder. But there is some optimism that we can make progress going down that path too, because there are significant conserved structure and so on in the spike proteins across coronaviruses. And so we're pushing on both of those aspects, though, both looking at just broad spectrum application of variants, but also then across the broader family of coronaviruses as well.
Thanks, Jim. Maybe one last question from Philippe, another technical question. There are hundreds of antibody, RBD and NBD structures now. Did you learn anything from the ensemble as opposed to optimizing one antibody?
Yeah, I think, we are constantly updating our models with new structures as they come out. So this is another area where structures are an important input to us. I don't know offhand how much the design processes have improved based on the ensembling of the multiple structural measurements that we have at this point. But I know that the team does actually bring those in and use an ensemble prediction of structure for that. What its impact is on the final result, I'm not quite sure. But again, I would be happy to connect you to the technical team to get into that in more detail, because again, these processes are open, and the publication should be out any day now too. I understand, so we're looking forward eagerly to that.
Yeah, thanks, Jim. I think sure. If you want to catch up for a coffee or something, we can give you a minute. And I would like to now invite Klas, and Jim, request you to stop sharing your screen.
Oh, sorry. Yes. Yeah.
And I think, Klas, you are going to run the panel now, so I leave it to your safe hands, and over to you. There are already questions coming for you in the Q&A tab.
That's excellent, and thank you, Sachin, and thank you, Jim, for a very interesting talk. Yes, if the panelists could turn on their cameras, I believe Inge, yeah, perfect. So here is the panel of today. You have met as an audience, you have met all of them earlier. They have all had great talks. So it's Randy Reed from Cambridge, Jim Brase from Atom and Lawrence Livermore, Jon Lærdahl, Oslo University Hospital and University of Oslo, Inge Jonassen from University of Bergen, and Victor Greiff from University of Oslo. I think we should, I will start by shortly take a round around the table here. If you could then say kind of what the last year, how has this impacted the work in general, but maybe also specifically your work. And I'm then thinking about the Rosetta Fold and AlphaFold version 2 that has came out. I also think that during the panel debate, if you just raise your finger or use your digital hand, I will pass the word on to you. But please, Jim, how has kind of the last events of the field, how has this impacted the field? And even more, how do you see the future here?
Yeah, I think that there have been a lot of exciting, new developments in sort of de novo prediction over the last year, and we've certainly been watching that and tracking it. I can't say it's really produced a lot of impact on what we're doing yet at this point, but I think the potential is really there to do this. I think because I showed some examples of the various ways that we use structure-based models in this. It's just a general comment is that that there isn't enough data to do this. Chemical space is too big, so using a purely data-driven models it's not going to get where we need, or allow us to accelerate this process enough. So bringing in structure information, using the basic physics and chemistry to supplement data that we have is going to be incredibly important. This issue of as we do molecular optimization, we move in chemical space. We can't go out typically and acquire 10,000 new data points from this. And in that yellow loop, we can get tens of data points or maybe 100 at the most. And we need to use structural information then to supplement that to build effective predictive models for the design process. So that's a general point. I think the things like the de novo prediction of structure in targets is critically important. We need to have it working well with co-structures of the targets plus ligands. And understand that well and understand its uncertainties also. And I think those are some areas that we're looking to see sort of continuing advances as we go. But it's a fast moving area and one that I think is going to have great impact. And it would be terrific to be partnering more on that. So that's a quick summary.
That's excellent. So the summary, you still have to spend your 1 million CPU hours per cycle to simulate this, or
yeah. Well, that's after we have the structures. So
exactly.
Yeah.
Yes. Let's move on, Victor Greiff.
Yes.
Same question.
Yes. So we are working on antibodies, and of course, antibody structure is very important for drug design. And there are many groups that are working on antibody structural prediction, and I see that they have already put out preprints, benchmarking their previous tools against AlphaFold now. So within weeks, these new benchmark preprints emerged already. So the field is moving very, very quickly now, and it will be very exciting to see to what extent we can use the principles that AlphaFold uses to bring them to the antibody field also. Where we have the problem that we cannot use the evolutionary information. So
exactly.
Yeah.
But in your methods, Victor, you also kind of simulate, like also spend a lot of CPU hours for that. Yeah. But you have also been experimenting, I think, going straight from the amino acid sequences to also binding problems, or?
Yes. Yeah. So we devised a simulation framework for antibody antigen binding, and I think in the future it will be nice to validate these in silico prediction using AlphaFold, for example. So that will be very nice. Yeah.
Excellent. And Randy, how do you foresee the future, and how has this kind of latest event impacted your work?
Well, I think the general trend in any technology that you use to address a scientific problem is that goes from being something where you need great expertise and lots of training to being something that's routine that everybody can do. And that was already in progress. This is just going to accelerate it tremendously. So instead of having to learn all of the tricks that you have for various difficult problems for how to solve crystal structures, which is what my group is specialized in, the answer is going to be press the button on AlphaFold or Rosetta Fold and do molecular replacement. And that's going to answer almost all questions. So great for science. It means we're having to learn new tricks and refocus ourselves a bit. But it's just going to accelerate the process of learning from structure and allow us to do things like you can do with the human genome, where you can mine large databases and find deep patterns in things. So.
Exactly. So what you say is that tools like, for example, ImmuneML, which Victor and others develop, those general tools, they will be applied more and more, so that you don't have to have kind of that much specific knowledge into the details to use them, and then these tools will provide you with kind of suggestions for drug design, maybe, or whatever. Yeah. Excellent. Inge, and you, how has this impacted you and your work?
Yeah, it is. So I'm not actively working myself in computational structural biology these days, but as a provider of infrastructure solutions for biological data, of course, this has a large impact for what data we can offer to the users out there, and what. So some of the things needs to be rethought a bit and be oriented in terms of research, as Randy talked about. So I of course know a lot about the research going on in the computational structure field, and that needs to be reoriented. And I think people are still thinking about how to best use the new wealth of information on structure. And this is also the case for the infrastructure and service provision side of this. How to best make this data available for users and integrate it with other resources and computational power and so on that is needed. I also work a bit on kind of personalized medicine and this kind of approaches, and there of course this will open new possibilities to use structure-based reasoning or tests where this has not been possible before. But I think we are just at the thinking about how to best exploit these resources.
So personal precision medicine is a keyword here, then. Jon?
Yes, for me personally, I've been invited to meetings like this and to national radio to talk about protein folding, which was new to me and to most people. Of course, I work mainly with in small projects with medical doctors, for example, who have patients with mutations, and they wonder which mutations are causing genetic disease. And in that case, it's very useful to have 3D structures of the proteins. Earlier, often I could not do any modeling. Now it's a larger percentage of proteins that can actually be modeled, so I can help more people, basically. And but it's only the last month, really, that the methods have been available to me, at least. So it will be very exciting for the next year and years to start using these methods.
And as we have seen, these tools also require quite a lot. I mean, it's not for anyone to do this either, although you will have more tools that are easily applicable in a sense. You still need the machinery behind that. So I also want to turn to Jim again. How is the Atom Consortium? I mean, how is this working together, both with the kind of academic institutions, national labs, and the industry? How is actually this collaboration going? It seems like both from DeepMind now and of course the Rosetta Fold also, these tools are open. So it seems like openness is the new standard in a sense. Is that also kind of what you feel in the Atom Consortium? And I mean, drug discovery and development also costs a lot. So the companies still need to earn their money. So how are you solving this? You're muted, Jim.
Yeah, there we go. Sorry. Yeah, solving it, I'm not sure, but that's a really important observation. Yeah, we know from our work over the years in AI that openness helps to accelerate complicated technical fields like this, you know, and AI machine learning is a good example of that, but it's been true in the computing area in general for a long time. Whereas in drug development, of course, the tendency has been and really continues to be exactly the opposite, to close everything in and protect it. So I really in the ATOM consortium, one of our, I think on one of the early slides, it said one of the goals was to democratize drug discovery. And I don't think I dwelled on that, but that is exactly what this is about. I actually think that both of those are important. At the early stage, I think openness is very important, to have a lot of what I would call shots on goal on these things, to have a broader variety of groups that have, cutting edge capabilities. And I think what we're doing in ATOM with doing a sort of public modeling and design optimization, and what's been done with AlphaFold of, doing very sophisticated protein folding and making it available publicly, it really helps support that trend. But then at some point, you're right, the requirement for money becomes so high that as you get to later stages of the process, it is important that you be able to protect the intellectual property that's being created here. What we're trying to do, though, is move that point further back in the timeline, so that the open groups, whether they're at universities, at small startups, in national labs, wherever, can get farther along the value chain there before they actually work with a specific company, say, and this becomes private. That reduces the risk for the company and adds the value to the front end groups. So that's sort of our philosophy in this, is it isn't to have to be open all the way through, but it's to move the boundary between open and closed further back in the process, which then expands greatly the number of groups and teams that can do that first part.
And do anyone else in the panel here have experience with the public-private partnerships and the tension that could appear between intellectual properties and openness and publishing? Randy?
Yeah, muted. Sorry, I was smiling because my one experience with a public-private partnership was one where all they wanted to know, they wanted a patent for something so that they could block anybody else from doing something.
Yeah.
So
that's the kind of thing we're trying to avoid. Yeah.
Exactly. Yeah, so that's absolutely what we try to avoid, and again, it was not obvious to me when DeepMind published their tool and we saw the results the 30th of November last year that these were going to be open. So my immediate thought was, when I saw the results, how could academia do this, or could academia do this through collaboration and as an open tool? And then the 15th of July, we kind of received the answer. Yes, academia could do this as an open tool, but still, also DeepMind did this and opened it. So if we go back to that question, is this kind of a trend? Do you think it will be easier to collaborate between academia and industry? Anyone has any opinions here?
Yeah, I guess my feeling is yes, absolutely. Having an open source tool base means that you can share things. So the idea, today with, just with research in AI in general, no one writes their own frameworks. We write it in TensorFlow, or we write it in PyTorch, or, or that, mainly those two. Yeah. So and that really facilitates collaboration, because then everyone's using the same frameworks, and you can share and work together at that higher level of abstraction. We'd like the same thing to happen in early drug discovery, where we have sort of common tool bases, common data sets that we can all use and explore methodologies and techniques. So yeah, I think creating the tool base that does that is really important, and that is one of the big goals with ATOM as well.
I think Inge had a comment.
Yeah, I completely agree with Jim. Having the common toolboxes and also more standardized formats and ontologies and so on enables industry to use code developed in the academia and vice versa. So I think the threshold for that kind of collaboration is much lower now than it was a decade or two ago. Then there was much less of this.
Victor?
Yeah, and I think also the funding agencies are, furthering this now with the Norwegian Research Council that likes to, that private and public cooperate together, and the EU via the IMI scheme, where we just got a grant funded on vaccine prediction research with GSK together, for example. So things are happening in that realm, I think, so which is positive. Yeah.
What do you think about kind of that trend that often then the public funding agencies like the Research Council, they may fund, for example, the infrastructure, which is used by academia, but more and more now also open to industry use and collaboration. Is that, we are touching upon the same question there, but that's a more and more obvious trend, I think, at least in Norway, but also in EU, these collaborations. So do you have any meanings or thoughts about this? Is it actually a public responsibility to fund these basic tools that you need to do these things now? Because you need a large infrastructure, you may need larger collaborations to do protein structure simulations or drug discovery now. Is it a public responsibility? Victor?
If I can, yeah. So I think as long as the companies are paying their share, it's okay, I think, right? And and I think with I think Google itself and also Microsoft and all the other technology players, they have their own server farms, right, which are much more powerful than what the public has access to actually in one entity. So I think one has to be careful, but if the private sector wants to participate in the public, then they should pay their part also, which I think they're doing, hopefully. Yeah.
Yeah. Inge.
Yeah, some of these approaches are very computationally demanding and resource demanding. So individual research groups on the bio side would not be able to set up DeepMind scale operations and AlphaFold and so on. So we need some common investments that makes these things available for the research community through research infrastructures and shared resources. That then we can justify the investments because they benefit so many groups. Without this kind of investments, the academia will lag behind, and it will only be happening in industry, these computationally demanding research, and that would really be a pity. So we need the academia also to be part of this. If it hadn't been for Rosetta Fold and so on competing with DeepMind, we don't know what would have happened with AlphaFold. We need a strong academic research activity.
And that's true.
Yeah.
And there you also, that's also a good example, because I mean the protein structures were provided through huge databases already for DeepMind to use. And is it then their obligation to kind of open their results, or in principle they would not have to do that? Of course, they probably saw some competition from Rosetta Fold and other tools, but it was not obvious at all that they were going to open it. Victor?
Yeah, I think that's a very contentious topic, and I think we can talk about this in general. But what I want to say is, of course, the Rosetta Fold and also AlphaFold progress is not de novo, right? This was decades of research, academic research published, right? So it's not that they came up with this in 2 years with some machine learning methods. Of course, the machine learning infrastructure that they're using and their software architecture is highly advanced. That's why it's also a bit difficult for academia to really re-engineer this. But and also I think if you want to publish in these higher journals, there's also via this preprint movement, right? That if you want to publish in Nature or Science, you need to open everything up. So, and since they wanted to publish in these high profile venues, I think there was the pressure from the public also that they make everything public also due to this preprint movement, which I think has greatly benefited science over the last few years, I think also. Yeah.
Also see a specific question to Randy Reed here. Is it not important to stress that experimental structures are still important? It is now easier to solve these structures.
Yeah, I probably didn't express myself. And first, hi Rick. Shared an office with Rick for 3 years back in the 80s. I wouldn't want to say that experimental structure determination is going to disappear. I hope I didn't imply that. It's just going to be a lot easier and the technical obstacles will be much reduced. But for the foreseeable future, I don't think we would want to make really serious decisions like designing a drug against something that you haven't actually experimentally determined or verified the structure of. So, but it's going to really accelerate the whole process, which is good.
Yep. You have your hand up, Victor. Is it? Do you have a comment? It's an old hand, the so-called old hands. Let's turn to more challenges. I mean, what are the challenges you see by this these new tools? We will have a lot of results, predicted results now. Any challenges around these predictions? We have seen a few. Maybe Randy could also answer that.
Challenges like
no, I mean. When it comes to predictions, that they are not necessarily what the world really is like. I mean, they are good predictions we have seen, but could also be erroneous. So
yeah, well, I do go on about how important error estimation is, but I think they've done a pretty good job of flagging up the things that you can't be confident of, especially with PAE matrix. But there are some challenges too, like if something isn't unfolded, it's a huge challenge to figure out, is there something else in the proteome that would make it ordered in a complex? And that's going to be really difficult to do computationally, I imagine, although I've been surprised before at what people can manage.
We have seen also throughout this workshop that things like protein-protein interactions are maybe the next step, antibody-antigen binding and so on. How do you see the development? What's kind of the next step going from what we now have, like structured tools, and into function drug discovery? We saw this also from your excellent talk, Jim. You have the whole infrastructure putting up this, starting maybe from structure and then ideally ending with new drug design that works, and also the acceleration that you talked about in the timelines here. So my question is really, what kind of what do you see as the next step to improve this process?
Yeah, from my point of view, I think one of the big challenges still remaining is translation of these new molecules into humans, because we even given good structural models with well understood errors and so on, and designing molecules that interact with those properly and so on, we still tend to be focusing on individual proteins in individual interactions in what is really a very complex network. And we ultimately need to really be designing, whether it's a molecule or a larger protein structure, we need to be designing them to understand how they perturb the overall network. And that's a very big challenge, I think, that for the future, because it's not, a typical small molecule in a real drug doesn't interact with one target. It interacts with tens or maybe even hundreds of targets. And understanding all of that and what the overall effect is, we're not even close to that today. So I think that's a remaining just outstanding challenge. And we won't get over there are two big problems in drug design. One is the time that it takes, and we're addressing that. And a lot of these tools are helping with that. And that's a good thing in and of itself. But the other problem is that less than 10% of these molecules that come out of that process actually make it through clinical trials and turn into real effective medicines. And that's really because what we're designing against is not a human. It's a very simple abstraction of a human into a single target or a small set of targets, or even an animal or something. They're not humans, and we don't know how to make that jump between to that level of complexity at this point. And as I said, that's still a long road to go, but a huge challenge for the future. So that's my dominant thinking.
Exactly. There is also a question to you about the ATOM project, and if you could say more about the ATOM on neglected tropical diseases.
Yeah, that's an area certainly of interest. I made a general comment that we want to apply this in areas of neglected diseases, things that pharma companies are typically ignoring. We don't have a program in that right now. We're still at fairly early stages of starting to really apply this. It's been, for the first 3 years, it's mostly been engineering development of this system approach, but we're starting that now. And if there are collaborators who want to come in and are interested in that area, we're pretty happy to work with people to apply these tools to different application areas. What's really required is some amount of data to get going, and you really need an effective set of assays to do this. But we'd be delighted to work with people in a lot of these open areas.
Do you? I mean, you have kind of this public-private collaboration, of course, already. But how do you see collaborating cross-country, also when it comes to not only drug design, but developing drugs, which means often patient data and sensitive data and so on? Is this something you're also doing within ATOM, or are you purely preclinical?
Well, we're mostly focused on preclinical. We have other projects that are addressing, patient data and so on. But in ATOM specifically, we're focusing on this preclinical aspect of it. Now, in the long-term problem I just talked about a minute ago, there will be needs to bring in, human relevant data, probably from the clinic as well, ultimately. There are obviously huge challenges. Our work with the Oslo Cancer Cluster has been an example of that. There are huge challenges in working with development of computational models of large-scale patient data. I've now spent the last year and a half in helping to manage the U.S. COVID-19 High Performance Computing Consortium. And we've supported over 100 projects. It's a consortium of government labs, academic labs, and industry. So all the big players, from IBM to Google to Amazon, Microsoft, and so on. But we've supported over 100 projects with free computing for this. And if you just look at the distribution of projects, the things that are focused around understanding of the virus, so molecular dynamic studies of proteins, early stage drug design, so virtual screens, AI-driven design processes, those are all working and producing, and there's good structure and support for those. The patient level projects are a disaster. We cannot share data. We cannot use data. We don't know how to do it. So it's just an area that needs a lot of work going forward, and something that group is actually quite focused on for the future. But we don't have a solution to that yet. It's a big problem.
Absolutely. Yes, of course. Also in Norway, we have a lot of public health data, and it's hard to work with as a researcher. Although things are happening, we are building a so-called health analysis platform in Norway, trying to make it easier accessible for research projects and also for industry, actually. There are many questions, so I will turn to a couple of questions from the attendees. One panelist question from Folmer Fredslund, mostly for Randy Reed, I guess. How might predicted structures influence the experimental structural biologist? E.g., will it be easier to identify appropriate targets, prediction of protein crystal packing, etc.?
I think there are some fairly obvious things like better construct design. If you know what are the where are the areas of flexibility in a protein, you might want to break it up. Where are the disordered N and C termini? You might want to chop those off. I'm kind of wondering what Jim was saying about picking the most informative experiment, whether there's a way of saying, is there a fold class where Rosetta fold and AlphaFold have the most difficulty in predicting it, and should we do a structure of that to fill in the data? So that would be an interesting thing.
Yeah.
But
so what you need, Randy, is maybe from the tools, computational tools, to try to see where they have the weaknesses and fill in those gaps.
Yeah. Sort of like a focused version of the structural genomics, which actually was another good example of public-private partnership in some cases. The Structural Genomics Consortium filled in a lot of the gaps.
Yeah. This is also, Jon, was a little bit about what you were talking about, new protein families and so on. But you were quite also surprised that you had good predictions also. Of course, some gaps, there are many gaps, but you were still surprised, Jon, about the structures.
Definitely. So I took some new proteins and impressive models, I think. But I think also with respect to drug design, these models that you get from AlphaFold, Rosetta Fold, they are, so the overall structure is impressive, but the exact details in active sites, good enough details to sort of computationally dock potential drugs in there, I think the quality is not good enough. So we need methods to, so of course, as Randy said, you need experiments to solve structures of the enzymes with ligands and drug candidates and things like that, but also computational techniques to optimize the details in parts of proteins.
So methods that kind of focus on the most important aspects of the proteins in a sense.
Yeah.
One question from Brian Edwards. If one has a drug target, how can AI naive user access the available open source programs and servers? Is there a recommended protocol? Anyone? If you have a drug target, how could you test it out? Could you use your immune ML, Victor, in the future, or how should you go about?
That's very much in the future, I think, this question. Yeah.
So we are still not there that you can test out drug targets in that sense.
I mean, it's a bit what Jim has been saying, right? And the other panelists also, we need better in silico oracles to score predictions and not go to experiment. Yeah, but we are not quite there yet, I think.
Yeah, I agree. I think doing it totally de novo, if all you have is the protein target, that's difficult still, and we're not there. Now, if you have some significant amount of data for molecules that have been assayed against this target at one level or another, then there are relatively straightforward, QSAR type methods that can be quite useful in this, and we use those extensively as well. So it really depends on your situation and what you know, how much data. But if you just have nothing but the target, that's a hard place to be right now. So then what you typically do today is you actually then run a physical screen against it, and that's what is commonly done. Purely virtual screens, I don't have a whole lot of faith in at this point. Same comment. So, but yeah.
We have a question. We have a question also related to this, Jim. If you could kindly share more information on open source platform by Atom.
Yeah, I don't think I showed the website. I can provide you. I can provide the group here with the website address and so on. I'll try to get it into the chat. If I can't do that, I'll get. I presume we're going to provide slides or something from this, and I'll make sure that slide is added to the deck because there normally is one in there, and I didn't include it this morning, that shows the website for getting access to those codes and data. So I'll provide that information.
Excellent. We will take a couple of more questions. Let's see, from Holmer Fredslund, protein structure prediction have definitely taken a big leap forward. This is within relatively few years. As we saw from Min-Kyung Baek's talk on Rosetta Fold, the ideas used in AlphaFold seem to be generally applicable by experts in the field. To the panel, within the few next years, what are the major advances, changes to science as you see it, based on the new availability of high quality predicted protein structures? And how much might the current algorithms improve during the next couple of years? I think this, we only have like 5 minutes left, so I think this is actually a very nice question to end with. So if you could take a round around the table with that final question. So Inge, please.
How much we can see improvement in CASP with AlphaFold 3 and so on, I don't really know. But I think using the techniques that have been developed on other related problems and working towards dynamics, for example, is really a new area that should be looked into. So at the moment we have predictive structures that are resembling the structures we see from crystallography, of course, which is the learning data, the training data. But proteins are not fixed structures; they are dynamic. So we need training data on dynamics and to use machine learning, artificial intelligence methods to predict the dynamical features as well. That way we can work towards understanding the machinery and the interactions, like Jim talked about earlier, to have a better tool to combat disease and so on. So that's at least one development that I would like to see, and that I think will be coming. And
you, Victor, what do you think? How much will the algorithms improve, and what's the development the next years?
Yeah. So if we believe, for example, of what science historians said about how science works, with first there's a paradigm change, and then you have opened a new door, and then there are many more people that can work on a new field. If we think about it like that we have now entered a new room, I think now many more people can put their hive mind together to kind of really improve those machine learning approaches in these cases, as John said, where we need higher accuracy. And as also Randy said, for these edge cases where we don't have so much accuracy yet, how can we improve those? So I think there will be a lot of improvement. But I think proteins, as was also just said, don't work in solitude. They like to interact with one another. So I think the next big thing is really, can we predict variant? So the impact of mutation on a structure level with respect to function. And I think both AlphaFold people and the Rosetta Fold people have said that this is one of their next big targets, which I think will be the next breakthrough if this can be done, for example, because then that would be the first thing where you almost don't need then an experiment anymore to really, if this is always true, then you don't need an experiment, wet lab experiment. Then you can just use the computer for variant function mapping. That would be unbelievable. Yeah.
Randy, do you agree?
Yeah, absolutely. I don't think I have much to add to that. Victor's talk went through a lot of the things that we should be looking forward to in the future.
And Jon, what about you?
Yeah, I also, Victor said it all. It's in a way a paradigm shift. I certainly didn't think we would be here a year ago. So it's difficult to see, to oversee what will happen in the future, but clearly clever people like Min-Kyung Baek took a look at the presentations, which I thought was not very clear. or from DeepMind at CASP 14 and was able to improve her methods by something like 10% from maybe 70 to 80 score. So clearly a lot of people are going to use these new insights in sort of clearly for fold prediction and also in other fields of biology.
And Jim, how do you foresee the future? Jim?
I'm sorry, I thought.
No, yeah, the same question. Where do you see how much do you think the tool will improve, and where do the next kind of advances come?
Yeah, I think I'll probably repeat some of Victor's points as well. I think there will be, I think one of the biggest changes immediately is just a change in the culture. You suddenly have all these AI groups everywhere interested in this. And that had been growing anyway, but this has really excited that community and made them realize that they could potentially do something important here, and that's a change, and a very positive one, I think, because that's the drug design ecosystem needed change. So I think that's a good thing. I think that the practical near-term thing will probably be that if we can actually get good starting point structures out of something like AlphaFold that are better than we can get today, that would reduce the need for this sort of initial data that we have and allow us to get started with things at an earlier point. And I think that's something we're pretty optimistic about, given what we've seen going on with this. I'm not quite sure it's there yet, but that's something we should be starting to try, I think. And then in the longer term, I think this whole issue about thinking of the target as a network effect rather than a specific single protein assay or something is really important. But that's probably a decadal project to work on. But that's where we're going to be going, I think.
I absolutely agree. And with that, I think we could end the panel discussion. I want to thank you all. And also, if I look at NORA and myself, I mean, I have no background in structural biology, but when these very exciting tools suddenly arrive from AI and kind of revolutionize the field, then you will suddenly have so much more traction. Many hands will kind of enter the field, also from AI background and other backgrounds, I'm sure. I think I will share a screen and do some final remarks and a short closing. Let me just share the screen first. So thank you to the panel again, this workshop was kind of made quite in a hurry, in a sense. After the July 15th paper, we saw that this is important. This is something we want to do. Also, the Norwegian community was very positive to this. In Norway, as Jim says, also with the Atom project and so on, we have big national structures. In Norway, we have a national infrastructure for computing, UNET Sigma 2, and also in collaboration with many of the big universities, we have infrastructure for do computing. We have also, like NORA, infrastructure for collaboration. And you see this big project now, like the Atom Consortium. These new methods, they require collaboration. So I think that's also a take-home message from this workshop. We need more collaboration, and we see that people are very willing to collaborate. People are sharing their tools. Open innovation it's even more open, I think, than it has been ever before. And also DeepMind opening their tools is also kind of an evidence of that. It has been great having these many people. As Jon also mentioned earlier, we were really planning this as a a bit smaller event. Then suddenly, I think it was Randy Reid that posted this to his network, and things really exploded. So this is just from yesterday. We had totally 759 people registered and 534 unique users attending the workshop yesterday. And this probably was added by some new people today. And as you can see, we reached out to most of the world, except for Norway, US, UK, Germany, Switzerland, France, and India were the countries that had many participants, and it's so nice to see that people are very interested in this field and from all over the world. We have gone through quite an extensive program with 4 sessions. First session was more fundamental on biology and also biotechnology. We have been through technical tutorials. We have also been through more kind of applications and limitations of these new tools. And in the last session now today, we try to look more on the future perspective, how to use these new tools when it comes to going from structure to function to drug design, and try to see a little bit into the future. I want to thank all the partners that has co-organized this event. University of Oslo, Center for Digital Life, Elixir Norway, Oslo Cancer Cluster, and D-Science at University of Oslo. A lot of people has been involved in both the planning and doing a lot of work for preparing this workshop. Special thanks to Sachin Gaur, Jon Lærdahl, Inge Jonassen, Jonas Verhellen, and Sabri Racek. You have tested the tools. As you all know, have seen, we have installed Rosetta Fold and AlphaFold version 2 at the machines in Norway. They are up and running for Norwegian researchers and students, but also for people abroad if you want to collaborate. As Sabri said, they are paid by Norwegian taxpayers' money, but anyone who wants to collaborate are welcome also to use this tool at the Norwegian computing systems. And not the least, thank you to all the speakers. It has been fantastic talks. We will put all and make all these talks available. So Sachin has already posted the links in the chat, and we will make them available there also for the future. You will suddenly probably also be able to download them and see them maybe through also YouTube channels. We'll see how to continue that. So with that, I think also I should, of course, say a big thank you to the teams that they have developed these great tools, like the DeepMind team and the Rosetta Fold team, who has made these tools open and available for the research community. This will surely be not only to benefiting the researchers, but it will be for the benefit for all of us. So thank you very much to everyone, and I hope you have enjoyed. Thank you.
Thank you. Bye-bye.
Bye-bye.